Okay, so the first thing our ship did on our incredible journey was we entered the outer cell membrane. And so my group determined that every time there was a shift from hydrophilic to hydrophobic and back, the ship was crossing a phospholipid membrane. So the first thing we did was enter the cell. And then the second thing we did was we enter the nucleus, because the nucleus is made up of two membranes, which means you're going to get the hydrophilic to hydrophobic blip twice, which occurs around between 2 and 3 on the graph. And then after that, in the nucleus, we entered the chromatin, and in the chromatin, DNA has the phosphate and carbohydrates, which accounts for those rises in the graph. And the sulfur is from the protein because of the disulfate bonds between the cysteine. And so then our ship goes at around 5, goes through a pore in the nucleus to the rough endoplasmic reticulum. And from 5 to 6 in the rough endoplasmic reticulum, sulfur exists due to ribosome-studded regions, and ribosomes are responsible for protein synthesis. So that's where you're going to get the sulfur, again, from those disulfide bonds between the cysteine. And there's no phosphate in that area. And then from 6 to 8, our ship enters a ribosome, which is going to show another sulfur peak since the ribosomes produce protein. And they're bound to the rough endoplasmic reticulum. Then from 8 to 9, you get a dip in pH, which means the solution is acidic. So from 8 to 9, the ship enters a lysosome. And here the macromolecules go through hydrolysis, which is going to cause the disulfide bridge to break, and we thought might expose sulfur more, which might cause the sulfur to rise. And then between 9 and 10, we exit the lysosome, and we exit the lysosome, so the pH is going to go back to a more neutral level. And then between 10 and 11 and a half-ish, we enter a mitochondria. So the big change in this part of the graph is that there's a big, the carbohydrate level rises and the phosphate level rises. So ATP has a triphosphate group, which is going to account for the rise in phosphate. And then mitochondria are responsible for cellular respiration. So that's going to account for the carbohydrate, because carbohydrates are what they break apart and help create ATP. And then at around 11 and a half, we're going to exit through the two membranes of the mitochondria. So that's going to account for the two hydrophilic to hydrophobic peaks. And then at around 12 and a half, our ship is going to enter the Golgi apparatus. And the Golgi apparatus is slightly acidic, which is necessary for protein modification. So that accounts for the pH dip at around 12 and a half. And then at 13, we're going to exit the Golgi apparatus. So that's another membrane cross, which is going to be a hydrophilic to hydrophobic little peak. And then we're going to exit the cell membrane. So that's going to be the last peak. And then, so my group decided to add an ATP sensor. And the ATP is going to exist in the mitochondria between 10 and 11. Like that. All right. That's it. Thank you.